Hello, everyone. My name is Bernd Meibohm from the University of Tennessee Health Science Center, and I will provide you some insights on the pharmacokinetics and pharmacodynamics of therapeutic proteins, now part 3. The goals of this section are to recognize the effect of major elimination processes on the disposition of therapeutic proteins, to understand the concept of target-mediated drug disposition and its impact on the PK/PD of many therapeutic proteins, and to appreciate the impact of renal and hepatic impairment on the disposition of therapeutic proteins. Therapeutic proteins undergo the same catabolic pathways as endogenous or dietetic proteins, and that means that they are degraded to amino acids as their building blocks that are reutilized in the endogenous amino acid pool. This proteolytic degradation occurs in a variety of different organs and tissues. Major sites of protein degradation are the gastrointestinal tract, the skin, the muscle, and the reticuloendothelial system. The main contributors are endothelial cells that use the process of endocytosis to take up vesicles with therapeutic proteins and then subsequently process those vesicles and degrade the proteins within them. Endothelial cells obviously line blood vessels throughout the body, and their overall surface area towards the blood vessels is more than 1,000 square meters in adult human. So even though the endocytosis process itself is relatively slow and inefficient, this is offset by the large surface area that's available to facilitate that process. That means that those organs and tissues that have large capillary beds with many endothelial cells are major contributors to the overall degradation of therapeutic proteins. And these are the skin, the intestine, and muscle tissues. In addition to that, the reticuloendothelial system, so mononuclear phagocytic cells like macrophages, monocytes, Kupffer cells, Langerhans cells, and others, are a major contributor to protein degradation by taking up therapeutic proteins, again, through vesicles into their in these cells, and ultimately the processing of these vesicles to degrade the therapeutic proteins that are included in them. Minor sites of degradation are also the liver, the kidneys, the spleen, and the lungs, but for specific therapeutic proteins, some of these organs may be a major contributor. The main prerequisite for proteolytic degradation is intracellular uptake. There are a variety of processes how this can be facilitated. The most basic one is called pinocytosis, which is a fluid phase endocytosis and the main process that endothelial cells use to take up therapeutic proteins. There are also receptor-mediated endocytotic processes, either by unspecific or by target receptors. Unspecific receptors are promiscuous membrane receptors like the LDL receptor, or sugar-recognizing receptors like the mannose or the fructose receptor that can recognize certain structural features in therapeutic proteins, and by that facilitate the endocytotic uptake of those molecules. Target-mediated endocytosis is then the binding to the pharmacologic target receptor, for example, a membrane-standing receptor that can then be internalized with the therapeutic protein receptor complex, and this complex can then internalized and further processed in the cell and ultimately lead to the degradation of the therapeutic protein. Target-mediated drug disposition is one feature that many therapeutic proteins exhibit, and that is basically the interaction with their target receptor that then also contributes an elimination process. So this is symbolized here in this little graphic where a pharmacokinetic two-compartment model is shown, where you have proteolytic degradation from either of those compartments. You can also have renal metabolism, and we'll talk about that in a second. And in either of these compartments, the therapeutic protein can now interact with its target receptor. And of course, target receptors also have a turnover kinetics, have a specific synthesis and degradation rate that ultimately influence and define the dynamics of this whole process. Now, once the therapeutic protein and the receptor interact, you get the receptor protein complex that ultimately drives the effect. This is usually defined by binding kinetics with a K-on and a K-off process. All this is similar to what we are familiar with for small molecule drugs. The major difference now is that this drug receptor complex or therapeutic protein receptor complex can be internalized in the target cell and can undergo lysosomal degradation. And by that, you not only have a receptor interaction that's reversible, but now you also have a unidirectional elimination process. So receptor interaction contributes substantially in this case to the overall clearance, and by that the disposition of the therapeutic protein. And since usually the target receptor is only available in a finite number on the molecular level, you can easily saturate that elimination process. And the consequence of that is a non-linear pharmacokinetic behavior. So target receptors are usually high affinity, low capacity binding sites. They're saturable at low molar ratios between the protein and the receptor. And that, as I mentioned, leads to dose-dependent nonlinear pharmacokinetic behavior. An example for that is a macrophage colony stimulating factor, which is eliminated by two parallel elimination processes. One is a linear renal metabolism process, and the second one is a receptor-mediated uptake into macrophages. So macrophages express the pharmacologic target receptor for MCSF. MCSF binds to that. The target receptor for MCSF complex is internalized and undergoes then lysosomal degradation. Since there is only a finite number of receptors on macrophages available, you basically get, with increasing doses, an increasing level of saturation of that elimination process. And that's shown in the graphic on the right side for three dose levels, 0.1, 1, and 10 milligram per kilogram. You see at the lowest dose level a very rapid decline in the concentration, since both elimination pathways, the linear metabolism as well as the receptor-mediated process, are fully active. So this is below the saturation level of the receptor-mediated process. When you go to higher doses, you start to saturate, to overwhelm the available receptors. So the receptor-mediated process gets to a saturation level. The extreme is shown here at the highest dose level, where you have now a much flatter curve, a much less or much slower decline in the concentrations, as now the process is largely only mediated by the renal elimination process through the metabolism of MCSF, but not by the receptor-mediated process. Once you get to concentrations that are low enough to get out of that concentration range, now the secondary parallel receptor-mediated elimination process kicks in, and you have the rapid decline in the concentrations. So this is a typical behavior that many therapeutic proteins exhibit. In a slightly different fashion, this dose-dependent clearance is now shown here in this graphic. On the y-axis, the clearance of the therapeutic protein. On the x-axis, the concentration in a logarithmic scale. Now again, I use a simple two-compartment pharmacokinetic model with two parallel elimination pathways, one through unspecific proteolytic degradation, that's non-saturable, or what I call a catabolic clearance, and then a second one that's receptor-mediated. And the receptor-mediated process can be conceptualized in a variety of different ways. One simplified way is shown here, where it is simply expressed as with Michaelis-Menten type kinetics as a saturable clearance pathway with a Km value that indicates the concentration at 50% of the maximum elimination and Vmax, the maximum elimination speed. And then the expression is shown here where the total clearance is the sum of the non-saturable catabolic clearance and the saturable target-mediated elimination pathway, conceptualized here as Vmax divided by the sum of Km and the concentration of the drug. So now in the graphic, you see the contribution to the overall clearance. The overall clearance is shown in blue. This has two components, the catabolic clearance in purple, the purple dashed line, which is independent of the concentration, but is at a low level. So it remains at this level. And then the second pathway, the receptor-mediated one, a high clearance level at very low concentrations. Once you get into a value around Km with the concentration of the drug, you get into the saturation range, and ultimately you can completely saturate that clearance process. And the sum of that is then the blue curve as shown here. So a high concentration dependence of the clearance process, depending on what kind of dose you give and what kind of drug concentration is still remaining in the body. That's the typical behavior that we experience with a lot of therapeutic proteins, and that is shown in a graphic here for a typical nonlinear pharmacokinetic behavior for monoclonal antibody that is given every 2 weeks. So you see the concentration time profiles that result from administration of either 200, 250, 500, 750, 1000, and 1500 milligram every 2 weeks. When you get to concentration levels that are much higher than the Km value or much higher than the saturation level for the target-mediated elimination process, you have nearly linear pharmacokinetic behavior. That's shown here for the 3 highest doses. Once you get to concentrations at the lower doses, where you get out of the saturation range for the target-mediated process, you get very rapid elimination at lower concentrations. And that is shown here in the drop, especially of the light green curve at the end of each dosing interval. So nonlinear pharmacokinetic behavior due to target-mediated drug disposition. Now, the kidneys can also contribute to the elimination of therapeutic proteins, but not as for small molecules by excretion, but rather than by metabolism. So therapeutic proteins are usually not excreted in unchanged form in urine, and if that occurs, this is usually a pathologic condition. So the kidneys, however, can be a major site of protein metabolism for smaller proteins that undergo glomerular filtration. Now, glomerular filtration is a rate-limiting step, and there's a size-selective cutoff of approximately 69 kilodalton, although the effective molecular radius based on molecular weight and conformation is probably more important and the limiting factor rather than the real molecular weight for the molecule. So glomerular filtration is most efficient if molecular weights are below 30 kilodalton. It reaches for very, very low molecular weights the glomerular filtration rate. But at higher concentrations, the filtration rate sharply falls off and has then this absolute cutoff of around 69 kilodalton. There's also a certain charge selectivity in addition to the size selectivity that may play a role. The consequence of that is that therapeutic proteins that are small enough to undergo glomerular filtration enter the primary urine, so the proximal tubulus, in the filtrate. And now they can undergo either interluminal metabolism through peptidases in the brush border membrane, and then the resulting amino acids and small peptide fragments can be taken up by peptide transporters, to especially peptide 2 and a lesser degree peptide 1, and can be reutilized in the endogenous amino acid pool. For larger peptides and proteins that are still small enough to undergo glomerular filtration, like interleukin 2, interleukin 11, growth hormone, or insulin, they basically are filtered into the proximal tubulus and are then taken up in these proximal tubular cells by endocytosis and undergo lysosomal degradation, again leading to amino acids that can be reutilized in the endogenous pool. There's also a third process that had been described, peritubular extraction, so direct uptake from the blood vessel, either by receptor or non-receptor mediated processes, which can contribute to the metabolism of therapeutic proteins like growth hormone and insulin, but usually is only a minor elimination pathway. So what does this then mean clinically with regard to patients that have renal impairment? For protein drugs with a molecular weight of below the cutoff of 60 to 69 kilodalton, I wrote here 60, I've had 69 on the previous slide. There's obviously, as I mentioned before, not an absolute cutoff in weight, but basically the hydrodynamic radius is more important. For regulatory purposes, the Food and Drug Administration usually uses the molecular weight of albumin with 69 kilodalton, as albumin is known to be not filtered by glomerular filtration. So for therapeutic proteins that are smaller than this cutoff, they are expected to be affected by renal impairment. There are examples out there that show this very nicely. One is shown here. That's recombinant human interleukin 10, which a molecular weight of 18 kilodalton, so it's below the cutoff, is filtered. And what you see here are concentration time profiles of interleukin 10 in patients with decreasing renal function measured as creatinine clearance, so either normal renal function, creatinine clearance larger than 80 milliliter per minute per square meter, and then successively reduced renal function. You see with reduced renal function, you get an reduced clearance, an increased systemic exposure, and an increased half-life of these therapeutic proteins. So what happens then on the other extreme with therapeutic proteins which have a molecular weight that's larger than the cutoff of 60 or 69 kilodalton? They are expected to be unaffected by renal impairment. Monoclonal antibodies, for example, with a molecular weight of 150 kilodalton are not filtered, and by that are not expected to be affected by renal impairment. An example is shown here with elotuzumab that has been investigated in patients with normal renal function, shown here as a solid line, compared to individuals with either severe renal impairment with a creatinine clearance below 30 milliliters per minute, or in patients with end-stage renal disease. And as you can see, the systemic exposure is very similar in those three groups. There were no relevant differences in AUC and Cmax between these groups, and by that, no effect on renal impairment on antibody disposition was observed in this specific publication. Now, since we talk about organ impairment, we probably should also mention hepatic impairment. So hepatic impairment, similar to renal impairment for those compounds that are larger than 69 kilodalton, hepatic impairment also has no impact for most therapeutic proteins. The liver is a major site of protein metabolism for some larger proteins, as I mentioned before, can be an important contributor in some specific cases. Studies in hepatic impairment patients are, however, rarely performed in drug development for therapeutic proteins, since hepatic metabolism is for most of them not the major elimination pathway, and no regulatory guidance by FDA is available beyond general recommendations. However, the colleagues at FDA put together a nice publication in 2020 where they collected data for 26 therapeutic proteins from their databases, mostly monoclonal antibodies, and they could show that in 23 of them, there was no effect of mild to moderate hepatic impairment on the pharmacokinetic that would require dose adjustments. However, there were a few exceptions. In 3 of them, there was actually a reduction in the systemic exposure, the AUC, by 35 to 70%. So it's basically the opposite of what you usually expect for small molecule drugs, where hepatic impairment of it oftentimes results in an increase in the systemic exposure. In this case, it was a decrease in the systemic exposure. And it's potentially related to concurrent hyperglobulinemia in hepatic impairment via saturation of the so-called FC RN or neonatal FC receptor process that we'll talk about in the next section. There's also a case report that I think makes the point very well for panitumumab, which has been used specifically for the treatment of a patient with metastatic colorectal cancer that had hepatic impairment, Child-Pugh class B, due to liver metastasis. And while small molecule drugs in this specific scenario could not be used anymore due to this hepatic impairment, this patient was specifically moved to a therapy with panitumumab as a KRAS wild-type anti-EGFR drug for KRAS wild-type metastatic colorectal cancer. The drug was well tolerated, and the concentration time profile observed in this specific case report was very comparable to what historic data showed in healthy individuals. This concludes the session on elimination processes for therapeutic proteins. In summary, unspecific proteolytic degradation is the major elimination pathway for most therapeutic proteins and is facilitated by endothelial cells and the reticular endothelial system. Target-mediated drug disposition may be a major contributor to the elimination of therapeutic proteins at low molar concentrations relative to their target abundance, and often results in nonlinear pharmacokinetic behavior. Classic elimination organs for small molecule drugs like liver and kidneys are only minor contributors to the elimination of most therapeutic proteins, and renal metabolism may contribute to the elimination of proteins with a molecular weight below 69 kilodalton that undergo glomerular filtration, and renal impairment may reduce the clearance of these proteins. Again, there are 2 self-assessment questions for this section.